I would like to introduce Dr. Carl Gutwin, who is going to talk about visualization for high-throughput phenotyping and genotyping. Dr. Carl Gutwin is a professor of computer science at the University of Saskatchewan, the co-director of University of Saskatchewan Human-Computer Interaction Lab, and a past Canada Research Chair in Next Generation Groupware. He has published more than 200 papers in human-computer interaction and computer-supported cooperative work. In 2012, he was inducted into the ACM-CHA Academy for his contribution to human-computer interaction research. Also, he was recognized as an ACM Distinguished Researcher in 2015. His research covers a variety of topics in human-computer interaction and computer-supported cooperative work, including interface design and organization, information visualization, modeling of human performance, collaboration support, and groupware usability. Ladies and gentlemen, please welcome Dr. Carl Gutwin.
The main things I want to get across to you about information visualization is that the idea is to offload cognitive processing onto our amazing capacities for visual perception. I also want to convey that each visualization answers a particular question, and so you should be thinking about what questions you want to answer. And then the mantra of InfoVis is overviews, filtering, and details on demand. So visualizations come in many sizes and shapes: winds in hurricanes, the best kind of dog to buy, the visual guide to the Big Lebowski for some reason, and apologies to anybody who's a Heather in the audience. This is a good example of using vision to think, because it shows a story about the amazing rise and precipitous decline of the popularity of the name Heather from about 1969 to 2001. This is probably the best infographic ever drawn. It comes from 1867, and by Sheryl and it describes, or it shows, Napoleon's march on Moscow. The only thing you need to know about this graphic is that it starts on the left-hand side in France, and on the right-hand side is Moscow, and the width of the line indicates the size of Napoleon's army. So you can ask yourself, well, what question does this visualization answer? And of course the question is, should I get involved in a land war in Asia? And of course the answer is no, you shouldn't do that. Okay, so on to the stuff that we've been doing in phenomics and genomic visualization for PERC. As you saw in the title slide, this is being done with a wide variety of people, a lot of collaborators who are giving us data, and a lot of students in Theme 3 who are putting these things together. So let's start with exploring plot data. And a lot of what we do in PERC is we work with images. And so those images provide us a base representation for overlaying other kinds of visualized information. This is picking up where Ian Stavnus left off yesterday. He showed you the process by which we gather the drone images, we stitch them together into Ortho Mosaics, and then split them back up into plots. This is a plot explorer that takes those split plot images and allows you to look at individual plots, look at the reps within a trial, and then once you put them down into drag them down to the bottom half of the screen, you can start to see the data that's been gathered about those individual plots, maybe from a machine learning algorithm or from a human who's gone out into the field and scored that variety. So we might be able to look here at 3 reps across one field and see what the differences are. We have 6 different reps, and then you might want to see, well, what are these varieties in comparison to one another as well? Now, these are just simple charts, but the idea is that we're trying to tightly tie together the phenotyping information with the data that we've gathered about that and put it all in one place. Okay, this is a front end onto that winnowing pipeline that Steve Siciliano talked about earlier this morning. And so the interesting thing about this is that, actually, Steve, I don't even know if is winnowing phenotyping or genotyping. I think it's somewhere in between. But the idea of this is when we watch people using this system, really what you do here is you set a bunch of the sliders and you set some parameters and then you press run. And what you're doing is you're exploring a parameter space. And what we realized is that people need some way of keeping track of what they've done inside that parameter space. As you all know, what you usually do is you just store a file that says, September 13th, Gutwin, do not delete version 2.txt, and then hope that you don't delete it. There are better ways to do this, and one of the ways is that we can track all of the versions that you use with this front-end system, and we can organize them in a tree because they tend to come from other versions. I changed this particular parameter so it's a descendant of this version. And it's very interesting now to see this as a base representation for helping people to understand how they've done their own scientific work. Okay, another one that comes from sort of the human side of trying to do phenotyping. If you remember the GoPro system, which is sort of that camera on a boom where you walk across the field, you might walk across this set of plots, and here's Liam walking down some, I think, canola plots, and he's going to go through six of these things. And the problem is that we end up with 300 images from each of those paths. And what we want to do is we want to turn those into an Ortho Mosaic, which means that we have to cut out all of the, just the pieces of that plot. This is the kind of problem that's easy for a human to do but hard for a computer to do. And what would be really nice is if we just had a big overview of the whole thing so that we could cut it out once. But of course, we're doing this in order to get an overview, so we can't do it using our main tools. So what we want to be able to figure out is how do we get a cheap overview? And so we're going to use this weird technique called slit scan photography, which has been around for ages but can be easily adapted to digital images. And we're going to take all of the images that we go across a plot, and we're only going to take the very central column of pixels from each one of those. And then we're going to mash them together, and we're going to get this weirdly interesting compressed version of the walk that Liam's done down that row of plots. And so if you take all of those central columns of pixels and squish them together, what you actually get is this, an overview that you were able to do within seconds of the entire run. And so it looks a little weird because we've taken that big set of plots and we've squashed them down in the horizontal dimension. Okay, and then we can just take a single operation and chop out all of the bits and pieces of the plot that we're interested in. And because we know where all of the original images came from in this overview, now we can go and cut those out automatically. Okay, so now moving on to genomics. There are also a variety of projects that we've been doing here, and data from a number of people who have done the assemblies and have been working with this stuff in PERC as well. Okay, so let's start with, this is the sort of the state of the art, or the thing that you'll find in most systems. It's JBrowse, and it allows you to go through a genome sequence and a bunch of other tracks. But this tool has enormous difficulties when it comes to remembering where you were, where do you want to go next, where have you been in the past. And so one of the things that we're looking at is alternate representations for specific genomes. And so here's an example of a browser for the NAPUS genome. And why not use the chromosome structure that you've got there? Why not use a gene density map to give you some visual indication of what that chromosome looks like and how it differs from other ones? This kind of a representation can provide you with an overview and can provide you with a much more memorable visual representation that you can use to hang your visual perception on and remember where you were. You can see some red marks in there, and we can also use this base representation to do things like visualize where you visited in the genome, and so that you can get back to previously visited locations relatively easily. Okay, another thing that we've recently been working on very hard is the idea of exploring synteny. And syntenic blocks are regions of a genome that are conserved either within the genome or across genomes and in hereditary lines. And this is now a popular thing to do, and it allows genome researchers to understand more about the conservation of traits and the way in which plant organisms have changed over time. So the first of these is SynVisio. This is done by my student Venkat. And so this allows you to compare a set of genomes on a number of different, with a number of different plots. This is NAPUS, and so there's enormous amount of conservation between the two parental lines within NAPUS. And so that's what you see there in that bar plot. And you can sort of switch on the chromosomes on and off, and you can see where those conserved regions of genes go to. You can filter by changing sort of the size of the block or the quality of the scan. This is coming from a bioinformatics program called MC-ScanX. The dot plot shows you in another form, if you have a line going diagonally down, that means that you've got a large amount of conservation between the two genomes. And of course, we can drill down and we can look at things in more detail. So sometimes the conserved region is flipped, and so you may want to look at that part in more detail. I believe that leads to some degree of difficulty in doing other kinds of analyses. And then you can go and actually zoom in and go and look at the actual individual genes that you may want to use for other analyses. Okay, so another plot that Venkat has built is called a Hive Plot. One of the problems with that earlier bar plot is that it can only be used for 2 genomes. And when you start to think about doing things like pan-genome analysis, there are 50 founder lines of canola. How are you going to show Synteny across 50 different things when you've only got 2 dimensions to show them? So this is one way of at least increasing that dimensionality to some degree. It's called a Hive Plot, and it allows you to look at several things as sort of spokes in a wheel. This, I believe, is the 3 of the wheat genomes that come out of Andy and Curtis's recent Nature paper, and they were kind enough to provide us with that data. And this shows some of the strong synteny between those different genomes, and I presume some of the interesting ways in which those things have differed or changed over time. Okay, another look at Synteny is through Jorge Nunez's work and his supervisor, Eric Neufeld. So this is a chord diagram. Some of you may know it as a Circos plot. It is quite popular in the genome community. And so this lays out chromosomes in a ring and then draws links between those chromosomes across the center of the circle. And so the interesting thing here is that this provides you with a different set of understanding of what these links are and can answer different kinds of questions about the genome. But as you can imagine, one of the big problems with these chord diagrams is that everything crosses in the middle. And so the problem of having a large number of these crossing blocks can occlude the analysis for the human. And so what Jorge has done is he's taken a computer science algorithm called simulated annealing and tried to come up with the best arrangement of these chromosomes around the circle that will minimize the number of crossings that exist in the diagram. And of course, you can go in and you can adjust. And of course, once you start doing this, it becomes kind of a game, and you lose the afternoon moving these things around. And then you realize, I'm not actually making this any better, and so then you ask the machine to do another version of it. So on top of all of these, other than the simulated annealing, some of these representations are not unlike what you see in the research literature, but the important thing is that we've now built them ourselves, and we have the opportunity now to take them in directions that nobody else has done. So visualizing other kinds of connections like the high C data from structural closeness that Steve Robinson has done, the pan-genome Synteny work, and the idea of tracing the evolution of a genome and being able to see the conserved regions and what happened to important genes and important regions as you go sort of through evolutionary time. And we can also put a variety of tracks on the outside of these plots as well. Okay, so I'm going to wrap up now, and the important thing is, I guess, is that some of these tools are online and some of them are coming online soon. You can look at these. These are sort of the prototype web addresses for these. You're welcome to try them out until you break them from overloading them. But the important thing is that you do have data that you want to visualize. Please do come and talk to us, because we're always looking for new questions to answer and new ways to look at things. Thank you very much for your attention.